Hello and welcome to another one of my You Ask and I Answer videos. And this video is in response to a comment I received on a video about genetic engineering and specifically about pooping pigs. So this was a video I have produced as part of a series of videos on genetic engineering, and you can find a link to the video in the description below and also above me now. So you can see the full comment on the screen now. I won't bother reading it, but what I'll do is I'll break it down and take it question by question. So the first question was, do you ever think about unintended consequences or that science has an incomplete picture that may be very inaccurate or fail at long-term extrapolations? So the simple answer to this question is yes, I do. If you look at one of my earlier videos in the genetic engineering series, you will see I pose the question, just because we can, should we? And I always think that should be at the forefront of our minds when we're doing any science, particularly something that could have long-term implications. So yes, I do think about the consequences a lot, about what could actually happen, what could actually go wrong. And yes, I fully accept that we have an incomplete picture of how biology works, how cells work, how the organisms work, how the ecosystem works and functions. But as scientists, we're always exploring that, we're building on our models, and we're constantly improving. So for example, when I started my PhD, this was our understanding of insulin signaling. Basically, insulin bound to a receptor on the cell surface, and then something happened. We understood about blood glucose levels going down and what would happen if the patient didn't have any insulin or the insulin receptor was faulty. We understood all of that, but we had no idea what was in this sort of mysterious black box between the insulin binding to the receptor and the physiological outcome. In the 3 years between me starting my PhD and finishing, we'd gone to a model that looked remarkably like this. And as you can see, in this model, it's incredibly complex. And even now, we don't fully understand this model. This image that you can see here, we produced a few years ago and is now out of date. And in this complex network of proteins, not all of which would be in any one given cell, but in this complex network of proteins that are involved in insulin signaling and connect in some way to the insulin signaling receptor, there is a fault that causes type 2 diabetes, and yet we still don't fully understand where that is. So yes, I fully accept that we don't understand how models work, and therefore with our genetic engineering we have to be careful and look at the intended outcomes or expect outcomes that we weren't expecting. So if you look at my genetic engineering playlist, you will see that in there I include examples of where genetic engineering has produced outcomes that were unexpected. For example, I talk about the use of CRISPR to genetically engineer babies so they don't have the CCR5 receptor and therefore are not susceptible to HIV. And in that work, the scientists found that they got off-target responses. They changed parts of the baby's genome that had unintended consequences, which we don't fully understand yet. So that's an example there. I've also included an example where I was working on a genetic engineering project where we were looking at rice and potatoes and how we again got an unintended or an unthought of outcome in that particular case. So have a look at those videos. The links for them are in the description below. But yes, the question, very valid point. We have to think long and hard about when we're doing genetic engineering. And I like to think that I do think about these things because it is of concern. One of the genetic engineering tools we have now is something called a gene drive, and that tool particularly worries me because once you release an organism that has a gene drive in it, there's no going back. That will go through a population. Have a look at that video and see if you can spot the problems that gene drives may have. So yes, think long and hard about any genetic engineering, and yes, we don't fully understand how things work. Now, the next question that was in the comment was, do you realise that most of science is actually flawed and just models of reality, but not reality itself? Yes, I fully accept that argument, and that's one of the reasons that I got into science, because I wanted to help with our understanding of these systems. Be it if our understanding is flawed, we design experiments to test our understanding and to improve our understanding. I mean, it's interesting when you look back through the history of drug design. Lots and lots of drugs have been designed over the years that are very successful with very few side effects. And yet when they were designed, We really didn't understand the underlying molecular mechanisms of how that drug worked or what it was doing to the cells, and yet we've got those drugs. But no, you're correct. We don't fully understand all the systems. We may never understand all the systems because there can be differences between organisms, and there can even be differences between humans with the way we metabolize drugs or the way we interact with particular compounds. So yes, fully accept your argument there. We don't fully understand all the models out there, all the systems that are out there. It is an incomplete picture. The next question in the comment was, how do you actually know if GM plants and animals is not creating 100 more problems than the problems they are solved by the act? Yes, you're right. We can have unintended outcomes. We can have things happen that we weren't expecting. For example, take a look at my video where I talk about the genetically engineered potato with reduced glycoalkaloid content. The experiments were designed, the work was designed to remove one glycoalkaloid, which could be involved in cancer. I mean, potatoes are perfectly safe to eat, but we were trying to make a potato that was potentially safer to eat. And what happened with that potato? We knocked out the glycoalkaloid, it was no longer made, but the potato replaced that glycoalkaloid with a different one. It turned on a gene. Now that new glycoalkaloid could potentially be more harmful. So that potato was never released, because we didn't fully understand at that stage what the glycoalkaloid was. We needed to do further work and look at it further. Now you could say, well, okay, why don't we just let potatoes get on with being potatoes? Well, if you look at any food crop that we have, they've all been genetically engineered. Sheep, cows, wheat, corn, barley, oats, corn on the cob, you name it, they've been genetically engineered because man naturally selects its best plants to produce a plant or an animal that gives the best yield. So we've been doing genetic engineering ever since we were farmers, because we'd always select, oh, that's our best potato plant, or that's our best strawberry plant, or whatever. So there is an element already in there of genetic engineering. We now just have the tools to do it quicker and easier in the lab without having to do all the breeding steps. Even with the sort of natural approach, it sometimes goes wrong. For example, look at my video on bananas. Here we've engineered a banana through classic breeding techniques. We've got a banana that travels well, is a nice fruit, is sweet and is tasty and is easy to grow. But we've ended up producing a banana that is in fact sterile. So now when a fungus has come along and causes that banana tree to die, we can't sort of cross back in protection against that fungus, because we've got a banana that's sterile. We have to use genetic engineering there to recover that banana. So yes, you can always have problems and unintended consequences and side effects, but we've got the science there and we will be looking for those and hopefully find them before they become a major issue. If it was something that was producing a chemical that was poisonous or something that was toxic, that would be picked up during initial screening of the plant, as we did with the potato example I gave you earlier. So yes, there could be consequences, but I think we have the tools and the methods in place to screen for those. The next question was, what if only those problems will be exposed after it's too late and the damage has been done? For example, what if GMing some living creature, it becomes wildly successful monetarily, but then after 50 years it turns out that the GM created an error in the genetic code causing the creature to produce a toxic substance that was causing cancer of all those that ate it? Well, again, that type of problem could occur not just as a result of genetic engineering in the lab, but through the natural selection processes that farmers operate. For example, there was a potato back in, I think, the 1970s that farmers had selected because it grew very well and it had a very long shelf life. What they didn't understand was it had a high glycoalkaloid content, and certain people, when they ate it, became very ill because they were very sensitive to glycoalkaloids. So it can happen with traditional breeding techniques. Again, I don't think this would be an issue because we have procedures in place for testing our GM crops and GM animals. Remember, in a lot of cases, we are just sort of tinkering around with one protein in these organisms or changing one thing. And with modern sequencing techniques, we can check that we've not altered anything else. It was through modern sequencing, for example, that with the CRISPR babies that I mentioned earlier, that we picked up that there were changes in other parts of the genome. So again, If we genetically engineer a change in an organism, we can go in and sequence the organism to make sure that we've only done that one change and we've not altered anything else. The next question was, does it matter to me? Well, yes it does, because I eat food, I live on this planet, so yes, I'm concerned about these things. But I think you have to come at these problems with a sort of open scientific mind and look at them rationally and sort of say, what is the risk here? What is going on? What are we changing? What do we know? Yes, we could get some unintended outcomes, but we've got the tools and the knowledge and the expertise to explore those and see what they actually are. So yes, it does concern me. Yes, it does worry me that people could do things without fully thinking them through. But science has got the tools to be able to deal with these sort of situations and to do the tests that we actually need to carry out. Why try to play God? No one is more intelligent than God or even comes close. Nature has evolved itself to work over billions of years. Yes, totally agree. Nature has spent billions of years evolving, developing animals. And farmers, as humans, we have spent millennia breeding animals, breeding crops to get the best thing. And that is genetic engineering, that breeding and selection process. In those situations, man, the human, is the selection pressure. We're the ones that sort of saying, okay, that pig over there is really good at producing meat. I'm going to cross it with that pig over there that's also good at producing meat, and we'll get a better meat producing pig. So there's selection there. It's just that it's being done by the farmer, it's being done by the human. And we now have the power to do that in the lab, to sort of cut out the long, sort of almost random step, because crossing those two pigs may produce a pig that produces better meat, but has some other issues. Classic example is look at dogs. Look at how humans have bred dogs over the years to select dogs for their traits, be it hunting dogs, guard dogs, or whatever. And as a consequence of that, we've ended up with dogs that have other issues. You know, some dogs are very susceptible to cancer or breathing difficulty or issues with arthritis and joints, etc. So with the old techniques, there are issues, as there are with the new lab-based techniques. The final question actually is, why do humans think they can come along and tinker with things and know the actual outcomes? Well, that's science. That's what we do. We tinker with things, we change things. If you look at the field of pharmacology, you're designing drugs there to treat illnesses, to treat conditions. Antibiotics are to kill bacteria so they don't kill us and infect us. And the same is true with genetic modifications. We're trying to improve things. We're trying to produce better crops, better animals, a better environment for us all to live in. So that covers all the questions that were asked in that particular comment, and I'd like to thank the commenter for their questions. As a lecturer, there's nothing better I like than receiving questions, because it means the person watching my videos is thinking about what I'm saying, is thinking about the science, and that is great. I used to really like it when I got questions back at the end of a lecture or during a seminar that made me think and made me question my own values. And these questions that I've just answered now have certainly done that. So thank you very much. Now, if you've liked what you've heard here and seen here, don't forget to like and subscribe. If you've got some questions, please feel free to leave them in the comments below. Bye for now.